Dr. Jaffe, can you explain what exactly happens when a lymphocyte shows a reaction in the LRA testing, especially in the context of photoactivation and the glycocalyx membrane?
I'm happy to give the headlines of what happens because there's a lot that goes on very quickly. First, we purify the antigen, we purify the substance being tested, we coat it on a novel microtiter plate. Think of a lot of dishes on one plate, a lot of wells on one plate, 48 actually, not 96. And we then bring the specimen up to room temperature and then up to body temperature. And we leave it there for 3 hours. And why do we leave it there for 3 hours? Because that's just the optimum amount of time for the APC, the antigen presenting cells, to pick up the antigen, process it, wiggle over to the lymphocyte, present the processed antigen at what's called the MHC locus, the major histocompatibility center, MHC locus. And what that means is it's like a key is inserted into a lock and the lock is opened. And when the lock is opened, a specific kinase. A kinase is an enzyme that adds phosphate molecules to proteins. So you make phosphoserine, phosphothreonine. And phosphate is very charged, highly negatively charged. So when the kinase turns on and adds phosphate to these glycoproteins, these proteins that have some sugar stuck on them, that are on the surface of the cell, those very surface glycoproteins get charged with phosphate and they push away the plasma and they push away from each other. And we see that under the microscope. And we know the difference between a true and a false positive reaction. We train our people to be very consistent. In fact, our reproducibility on blind split samples is less than 3% variance, which is exceptionally good for any lab test and for a cell culture gold standard. Now, that same kinase, known today as a JAK2 kinase. Why? Because the biochemist named it JAK2. That same kinase sends messages to the nucleus to start dividing. So if you waited 2 days, notice I said we wait a few hours and then we read the cell response. If you waited a few days and you looked at incorporation of, say, thymidine, you could see that cell division stimulated by that phosphorylation was underway. And if you waited 3 weeks, as used to be done in the laboratory, you could actually grow a colony of cells. So there's one specific lock that is opened by a presented processed antigen. We measure the results of that through this kinase system, and we measure it early rather than late. So if you looked at colony formation, the best you could do was plus or minus 30% variance. If you looked at thymidine incorporation, the best you could do was about 15 to 20% variability. Looking the way we look at the entire family of cells and how they react in multiple parts of the reaction chamber. So we don't just look at one little spot, we look at several spots. And it is a qualitative endpoint. If most of the cells light up, that's a strong reaction. If some cells light up, that's a moderate reaction. If no cells or a very rare occasional cell react, that's a non-reaction. And we're often asked, couldn't we quantify the kinase activity? We might. It would cost an awful lot more. We might be able to quantify the kinase, but it would be a difference without a meaning. Because then I think you would ask me, is an 88 kinase activity score different than a 68 or a 51? Not that that we know. It's a highly sensitive, high amplification, very reproducible system that we have been offering clinically since 1983. We're over 90,000 specimens, we're over 27 million cell cultures. And so we are quite consistent as the standard against which lymphocyte response assays, immune memory lymphocyte assays, T cell and B cell assays, are performed clinically.